Now let us look at the topic inoculum development, and we'll also look into scale-up fermentation in this. Okay, so what is inoculum development? Inoculum development can be defined as the preparation of population of microorganisms from a dormant stock culture to an active state of growth, that is logarithmic culture, that is suitable for inoculation in the final production stage. And this is called as inoculum development. That is, you are preparing a microorganism from the stock culture and changing the stock culture, which is dormant, is converted into actively growing stage, which can be used for the production in the bioreactor. So this inoculum development, it is a stepwise process. Okay, and in this inoculum development, you will be increasing, a gradual increase in the inoculum will be done, or gradual increase is called as a scaling up. Scaling up of the volume of inoculate will be done up to the desired level. So the inoculum, what are the importance features of the inoculum that can be used for industrial fermentation? The inoculum or the culture that is used for industrial fermentation should be in its active state. It should be healthy and in the exponential or logarithmic phase of growth. It should be free of contaminant and it should be available in large volumes, because the bioreactor is like thousands of liters. The inoculum should also be in large volume, and the inoculum should be able to retain its capacity to for the formation of the desired product, so that there is no genetic variability, no chemical variability or whatsoever in the inoculum. So these are some features which the inoculum should be having. Okay, so there are like a few steps for the inoculum development. The very first step is the inoculum is taken from the working stock, okay, to initiate its growth into the liquid medium. So you'll be having a stock culture which in which your inoculum is maintained in its dormant form or in its less active form. So from this stock culture, working stock culture, you'll have to initiate the growth of the organism. So you'll be inoculating this in a suitable medium, okay, in a liquid medium. And how is it done? The bacterial cell, the vegetative cells and spores can be suspended in sterile water, sterile tap water, and this suspended sterile tap water or this liquid can be then added to the culture media or the broth. In case of non-sporulating fungi, so if it is vegetative cells of bacteria or spore, you can do it in the sterile tap water. Or if it is a non-sporulating fungi or actinomycetes, what we do is we'll take the hyphae of these organisms, fragment it, break it into small pieces, and then transfer it into the broth. So from the stock culture, you will be adding it to broth. Then next, the inoculum is development is done using flask culture of 50 ml to 12 liter, or a small fermenter is then used. So initially you are using this inoculum and you're growing it into a very small test tubes or vial, and from this you will make like a into a bigger flask. From the small vial you'll transfer it into a bigger flask of 50 ml up to 12 liters of medium. That is a small fermenter, a laboratory fermenter of 12 liters. Then this way, like we are doing, like from a very small volume to a flask to a little fermenter, it is done in a stepwise sequence by increasing the volume up to the desired level. So if the final volume that should be incorporated into the fermenter, the final volume of the inoculum is 30 liters. From this 12, you will be going into a bigger fermenter, and you will be maintaining a 30-liter volume of inoculum. You will be growing it, making it into logarithmic phase, and this 30-liter will be inoculated into the medium. So at each step, what are you doing? You are using 0.5 to 5 percentage of inoculum and adding it into the medium. And in this medium, it is allowed to grow to increase 20 to 200 fold by division. And when this medium is like, when it is growing well, it is taken to the next volume. Okay, so at the production stage, again 5% of the total medium volume will be taken. So if it is a 1000 liter fermenter, 1000 liter fermenter, you will be taking 5% of 1000 liter, that is 50 liters of inoculum. And the thing here is, whatever medium you are using for inoculum development, for step by steps scaling up of the inoculum, whatever medium you are using will be very similar to the composition of the medium in the fermenter. So this similarity will prevent a lag phase when you're adding your media or your inoculum into the medium. If you're growing the medium in one particular broth and the fermenter is having an exactly like a different environment, different culture setup, the organism will take time to adjust in the fermenter. So to overcome this, you'll be taking even inoculum development and the fermentation medium will be very similar. So this is a setup of inoculum development. You can see that from the culture, okay, from the slant or the cryopreserved culture, you'll be adding a little into a small um, LMAN flask. From this, you'll go on, you'll add it into, you'll grow it, you'll add on it into a bigger one, and then bigger until it reaches the fermenter.